3D printing of biocompatible polymers. Materials, additive manufacturing technologies, challenges, and future perspectives. Three-dimensional, 3D printing, also known as additive manufacturing, has revolutionized biomedical engineering by enabling the fabrication of complex three-dimensional structures directly from computer-aided design (CAD) models. Unlike conventional subtractive manufacturing, which removes material through machining or molding, additive manufacturing constructs objects layer by layer, allowing exceptional control over scaffold architecture, pore geometry, internal channels, and external shape. This design flexibility has made 3D printing one of the most promising manufacturing technologies for regenerative medicine, tissue engineering, drug delivery, and patient-specific medical devices. The ability to fabricate customized implants that match a patient's anatomy while incorporating living cells, bioactive molecules, and biodegradable polymers has transformed the development of next-generation biomedical materials. The success of biomedical 3D printing relies heavily on the availability of biocompatible polymers that provide both structural integrity and biological functionality. An ideal polymer should possess excellent cytocompatibility, controlled biodegradation, suitable mechanical strength, sterilization, stability, and printability while promoting cell attachment, proliferation, and tissue regeneration. Consequently, considerable research has focused on developing natural and synthetic polymers whose physicochemical properties can be tailored to satisfy the demanding requirements of diverse biomedical applications. Biocompatible polymers for 3D printing. Biocompatible polymers employed in additive manufacturing can generally be classified into naturally derived polymers and synthetic biodegradable polymers. Each class offers distinct advantages, and increasingly, hybrid polymer systems are being developed to combine their complementary properties. Natural polymers are attractive because of their close resemblance to the extracellular matrix, ECM, the highly organized network of proteins and polysaccharides that supports cellular functions in living tissues. Materials such as collagen, gelatin, alginate, chitosan, cellulose, hyaluronic acid, fibrin, silk fibroin, and dextran inherently promote cell adhesion, proliferation, and differentiation due to their biological origin. Their hydrophilic nature facilitates nutrient transport and cellular infiltration, making them particularly suitable for soft tissue engineering and bioink formulations. Among these materials, collagen remains the gold standard because it is the primary structural protein of mammalian connective tissue. Collagen-based scaffolds closely mimic native ECM architecture, providing biochemical signals that regulate cell attachment and tissue remodeling. However, pure collagen exhibits relatively poor mechanical strength and rapid enzymatic degradation, necessitating chemical cross-linking or blending with synthetic polymers. Gelatin, a denatured derivative of collagen, retains many of collagen's biological properties while exhibiting improved processability. Methacrylated gelatin, GelMA, which undergoes rapid photocross-linking under ultraviolet or visible light, has become one of the most widely used bioinks for stereolithographic bioprinting because it combines excellent cytocompatibility with tunable mechanical properties. Alginate, a naturally occurring polysaccharide extracted from brown seaweed, is another important hydrogel-forming polymer extensively employed in extrusion bioprinting. Rapid ionic cross-linking with calcium ions allows alginate hydrogels to solidify immediately after deposition, enabling excellent print fidelity and encapsulation of living cells. Nevertheless, alginate lacks intrinsic cell-binding domains and is therefore frequently blended with gelatin, collagen, or nanocellulose to improve cellular interactions. Chitosan, derived from the deacetylation of chitin obtained from crustacean shells, offers unique antibacterial properties together with excellent biodegradability and biocompatibility. Chitosan-based scaffolds have been widely investigated for wound healing, cartilage regeneration, drug delivery, and antimicrobial tissue engineering. Similarly, nanocellulose has attracted considerable attention owing to its remarkable mechanical reinforcement capability, shear thinning rheology, and renewable origin. Cellulose nanofibers significantly improve printability while simultaneously enhancing scaffold stiffness without compromising cell viability. Although natural polymers exhibit excellent biological performance, their relatively poor mechanical strength, rapid degradation, and batch-to-batch variability often limit their application in load-bearing tissues. Consequently, synthetic biodegradable polymers remain indispensable for biomedical additive manufacturing. Synthetic polymers provide greater control over molecular architecture, degradation, kinetics, crystallinity, mechanical performance, and processing characteristics. Among these materials, polylactic acid, PLA, is one of the most extensively utilized thermoplastic polymers for fused filament fabrication. Derived from renewable resources such as cornstarch and sugarcane, PLA exhibits excellent printability, moderate stiffness, and complete biodegradability through hydrolytic degradation into lactic acid. Because of its ease of processing and relatively low melting temperature, PLA has become the preferred material for manufacturing customized orthopedic models, biodegradable fixation devices, and porous bone scaffolds. Polycaprolactone, PCl, has emerged as another important biodegradable polyester owing to its low melting temperature, approximately 60 degrees Celsius, excellent flexibility, and exceptionally slow degradation rate that may extend over several years. These characteristics make PCl particularly suitable for long-term tissue regeneration applications requiring prolonged mechanical support, including bone, cartilage, tendon, and nerve regeneration. PCl is frequently combined with hydrogel bioinks, where it serves as a mechanically robust framework surrounding softer, cell-laden materials. Polyvinyl alcohol, PVA, represents one of the most versatile hydrophilic polymers used in biomedical engineering. Unlike thermoplastic polyesters, PVA readily forms physically or chemically cross-linked hydrogels that closely resemble the viscoelastic properties of native soft tissues. The polymer exhibits excellent biocompatibility, high water retention, oxygen permeability, chemical stability, and outstanding compatibility with numerous natural polymers, including gelatin, collagen, alginate, and chitosan. These characteristics have led to extensive investigation of PVA-based hydrogels for cartilage repair, vascular grafts, artificial corneas, liver tissue engineering, wound dressings, drug delivery systems, contact lenses, and soft tissue implants. Furthermore, PVA exhibits tunable swelling behavior and mechanical properties that are particularly advantageous for pelvic floor reconstruction and abdominal hernia repair meshes, where flexibility and tissue integration are essential to minimize inflammatory responses associated with conventional polypropylene implants. Other important synthetic polymers include polyethylene glycol, PEG, polyethylene glycol diacrylate, PEGDA, poly-beta-amino ester, cheese, PBAES, polyurethane, PU, polyetherether ketone, PEEK, and polyvinyl pyrrolidone, PVP. PEG-based hydrogels have become widely adopted because of their exceptional hydrophilicity, non-immunogenicity, and chemical versatility, allowing facile incorporation of bioactive peptides and growth factors. polyurethane provides excellent elasticity for cardiovascular and soft tissue engineering, whereas PEEK offers outstanding mechanical strength and chemical resistance for permanent orthopedic and spinal implants. Modern biomaterials research increasingly favors hybrid polymer systems that combine natural and synthetic polymers to achieve high biological activity and desirable mechanical performance simultaneously. Composite formulations reinforced with nanomaterials such as graphene oxide, hydroxyapatite, bioactive glass nanoparticles, carbon nanotubes, or nanocellulose have demonstrated significant improvements in tensile strength, osteoconductivity, electrical conductivity, and cellular response, thereby expanding the functional capabilities of printed biomedical constructs. Additive manufacturing technologies. The successful fabrication of biocompatible polymer scaffolds depends not only on material selection, but also on the choice of additive manufacturing technology. Different printing methods operate according to distinct physical principles, and therefore require polymers with specific rheological, thermal, or photochemical properties. Fused filament fabrication, FFF, also referred to as fused deposition modeling, FDM, remains the most widely accessible 3D printing technology due to its simplicity, affordability, and compatibility with numerous thermoplastic polymers. In this process, a continuous polymer filament is heated above its melting temperature and extruded through a fine nozzle to produce successive layers that fuse upon cooling. Materials such as PLA, PCl, PEEK, and thermoplastic polyurethane are commonly processed using FFF. The technique enables the rapid fabrication of mechanically robust scaffolds possessing highly controlled pore architectures and interconnected porosity, making it particularly suitable for bone tissue engineering and patient-specific implants. However, the elevated extrusion temperatures generally preclude direct incorporation of living cells, restricting its application primarily to a cellular scaffold fabrication. Stereolithography, SLA, represents one of the highest resolution additive manufacturing techniques available for biomedical applications. SLA selectively polymerizes liquid photopolymer resins using ultraviolet or visible laser irradiation. Successive exposure of individual layers generates highly precise structures with smooth surfaces and micron-scale resolution. Closely related digital light processing (DLP) technology employs digital projection instead of laser scanning, allowing entire layers to be cured simultaneously and substantially increasing printing speed. Photocurable hydrogels such as Gel-MA, PEGDA, and methacrylated hyaluronic acid are particularly suitable for SLA and DLP because rapid photopolymerization preserves complex geometries while maintaining high cell viability. These technologies have become indispensable for fabricating microvascular networks, organ-on-chip platforms, and highly detailed tissue scaffolds. Selective laser sintering, SLS, utilizes a high-energy laser beam to selectively fuse polymer powders layer by layer. Unlike extrusion-based methods, SLS requires no support structures because unsintered powder naturally supports overhanging features during fabrication. This advantage enables production of highly porous structures with exceptional geometric complexity. Biocompatible polymers, including PCl, PEEK, nylon, and polymer ceramic composites, have been successfully processed using SLS for orthopedic implants, craniofacial reconstruction, and customized prosthetic devices. The technique also permits incorporation of ceramic fillers, such as hydroxyapatite and beta-tricalcium phosphate, producing composite scaffolds with mechanical properties approaching those of natural bone. Direct ink writing, DIW, also known as extrusion bioprinting, has become the dominant technology for biofabrication because of its compatibility with cell-laden hydrogels. In DIW, highly viscous bioinks are extruded through micron-scale nozzles under pneumatic or mechanical pressure. Successful printing depends critically on the rheological properties of the bioink, particularly shear thinning behavior, yield stress, and rapid structural recovery following extrusion. Shear thinning hydrogels decrease their viscosity during extrusion, minimizing mechanical damage to encapsulated cells, while rapidly recovering viscosity after deposition to preserve scaffold geometry. Materials such as alginate, gelatin, collagen, gel-MA, chitosan, and PVA-based hydrogels are extensively employed in DIW for constructing vascularized tissues, cartilage, skin substitutes, and organoids. Inkjet bioprinting represents another important technology that deposits microscopic droplets of bioink through thermal or piezoelectric actuation. This non-contact approach enables high printing speed and precise spatial placement of multiple cell types and biomolecules. However, the technique is generally restricted to low-viscosity bioinks, while repeated droplet ejection may expose encapsulated cells to thermal and mechanical stresses that reduce viability. Despite these limitations, inkjet printing remains valuable for fabricating highly organized multicellular tissues and high-throughput drug screening platforms. Recent advances have introduced volumetric bioprinting, free-form reversible embedding of suspended hydrogels, fresh, and multi-material printing, enabling fabrication of complex tissue architectures with unprecedented speed and structural fidelity. These emerging technologies significantly expand the capability to reproduce the heterogeneous organization of native biological tissues and represent important milestones toward whole organ fabrication. Overall, the synergy between advanced biocompatible polymers and increasingly sophisticated additive manufacturing technologies has positioned 3D printing at the forefront of regenerative medicine. Continued innovations in polymer chemistry, bioink formulation, scaffold design, and printing methodologies are expected to overcome existing limitations and accelerate the translation of 3D printed biomedical constructs from laboratory research